Hello, everybody. My name is Paul Hacker. I am a PhD candidate at UBC, working under Dr. Nicholas Coops. I am 3.5 years into my program. So today I'm going to talk to you about the last 3 of my 4 research questions that are going to go into my dissertation. Essentially, I'm working to evaluate biodiversity in dairy oak ecosystems, which are mixed woodland, grassland savannas, and we're using leaf and drone-based imaging spectroscopy. Little overview of the site. We did all of our leaf spectroscopy in May 2019 during the prime bloom season for the meadows. Essentially, we collected a variety of things. We sampled 23 plant species, between 6 and 10 samples of each species, conducted leaf spectroscopy between 400 nanometers and 2500 nanometers, derived 14 leaf chemical traits. That was all done at UDM, and also collected VisNear with the UCASI and VisWeir imagery from drones. Thank you to Margaret and her crew for that. An important part about this site is, I guess, its relation in British Columbia and in Canada. It's one of the most endangered ecosystems in Canada, and it includes a variety of endangered species, not just plants, but animals as well. And it experiences a variety of stressors. Each of these that I've put up here, whether it's agriculture, transportation, and the road system, or encroaching coniferous forest, all of those threaten this continuously diminishing ecosystem. And that's actually slightly ironic because with climate change, its range is expanding. So you wouldn't expect it to be diminishing, you would expect it to be growing. So the first research question that I'm going to talk about has to do with spectral mixing. So essentially, what are the effects on trait prediction if we observe multiple different plant species in the same pixel? I should give you a basis of where this idea came from. The thought was that if in the same pixel you had one species with a high leaf nitrogen content and another with the low leaf nitrogen content, the value you would predict from the spectra could be in the middle. It would be a mean, and that really doesn't capture the function. So, are you able to say that this is the actual functional trait value of this pixel when you have such a disparity? And maybe you can, maybe you can't. That's what we wanted to find out. Before we could do that, obviously we had to do some functional trait modeling. I talked with Sean to do this, so he's outlined the methods pretty well earlier, and similar results that we found. Leaf nitrogen percent, the carbon nitrogen ratio, LMA, LDMC, all predicted relatively well. And overall, we could accept 8 of the traits. So you can see an asterisk beside the accepted traits, which were then evaluated. So there are 3 hypotheses with this research question. The first, spectral mixing increases the prediction error of functional trait estimation. Essentially, if there's more than one species in a pixel, you're going to predict the wrong trait value. Hypothesis 2, traits are not equally affected by spectral mixing, so perhaps chlorophyll A is more affected than LMA. And hypothesis 3, spectral mixing with different plant life forms does not affect trait prediction. So we thought that it shouldn't matter if there's a herb, if there's a forb or a graminoid or a shrub, they should all be affected similarly, and explored if that was true. Sort of a key part of this was creating the fractionated spectrum, because we were using the leaf spectra that Sabrina and the crew collected in 2019. And so the first thing we had to do to test this was select a target species of which to evaluate the changes in the predicted trait with its pure end member predicted trait. And we settled on the species Vicia sativa, which is a common vetch. It is native to Western Canada. I don't know how much in the rest of Canada, but essentially it occurred in 100% of the plots that we measured, from as low as 1% cover to as high as 30% cover. So it's incredibly popular. And it has an interesting growth strategy. It essentially curls around a leaf, so its leaves will be on top of the leaf that you're observing. And so it seemed like a prime candidate to test if this mixing happened. Ultimately, the creation of fractionated spectra was really just taking the end member spectra and multiplying it by the percentage that it would represent in the mixed spectra. So If an n member was only 75%, you would multiply each n value by 0.75 and get the fractionated spectra. From that, we could mix, or we did mix 2, 3, and 4 species mixes, and you really take those sort of Frankenspectra, if you will, and use the models that we created for each trait to predict the mixed trait. So this graph is referring to hypothesis 1, and I'll explain it a little bit first before I talk about the actual results. So starting on the right-hand side of the graph is 100% target species inclusion. So you can see here we have this red line that goes all the way across for LMA, so 37 grams per meter squared, and the dotted lines are the 95% confidence interval. So what we're looking for is to see if, as we include another species from 2 to 4, 2, 3, and 4, how much does our error increase around the true prediction of Biodiversity sativa? You can see for LMA, it actually remains relatively constant. There are some outliers that go down below, and interestingly, there is one that consistently increases as its presence increases as well. So there are some interesting things looking at that graph, and again, LMA tends to hold relatively well regardless of mixing for most species. These two graphs sort of depict what we were talking about before, where you would, if you had multiple species that had a very different trait value, you would end up getting an average. And so these graphs refer to whether or not traits are affected equally. And you can see even with the average percent change here, there are some that are negatively affected, that sort of decrease in no change, the black line is, and some that go above. But in general, the amount that they change between 2, 3, and 4 species is not that different. And so what you need to look at is a cumulative percent change, where you start seeing, as you add another species in, you start getting 2, 3, 4,000% change in the total trait value. So that is sort of confirming that regardless of the average not changing that much, when you do add more species, you do add more interactions that could be higher, lower, and again resulting in that incorrect averaging. And hypothesis 3 dealt with the effects of mixing with different life forms. So you can see we have 4 graminoid, shrub, and tree. The tree, there's only one tree species that we included in this analysis, the Carcassariana, which is Gareo, flagship species of the ecosystem, but for us it was. And it's just represented by this single line right here. And you can see, as I mentioned earlier, for LMA, even when you're just breaking it into life form, pretty much is the same exactly what we saw when you had 2, 3, and 4 species mixes, and it was in fact a tree that had a higher LMA, which is not surprising. But it's interesting to note that as soon as it enters at, whatever that 90, even if it's just 10% in the pixel, you're going to be able to say, oh no, this is not Biodiversity sativa, and then you can see it's much greater than the other species as well, regardless of their life form. Nitrogen percent was another interesting one, where we actually had almost all species of all life forms decreasing from Biodiversity sativa, which is not surprising necessarily. Biodiversity sativa is a nitrogen fixer. But there's another nitrogen fixer which has a much more ominous ecological presence in Gareo ecosystems, and that's Scotch Broom. And so you can see here, as its presence increases, it's quite detectable. So a quick overview of what we actually found. Hypothesis 1, trait prediction accuracy does change due to spectral mixing. Hypothesis 2, certain traits are more effective than others. An example of that was that nitrogen percent was more effective than chlorophyll A, and there were some other key differences like that. Hypothesis 3, that mixes containing essentially various percentages of different life forms, they do present different prediction errors in some of the traits, including nitrogen percent. But for the most case, leaf mass per area was relatively stable unless Carcassariana was included. So again, a potential method of identifying some tree crowns in this ecosystem. This is manuscript is completed and is currently under review at Ecological Indicators. Third research question, as I mentioned, the very evil Scotch Broom. Why, Paul, is it evil? Some might ask. Thank you, Nicholas, for asking that question. It's very insightful. And so Scotch Broom is originally from the Mediterranean and essentially was considered a fine ornamental shrub and carried throughout the New World. So it's colonized now in New Zealand, Australia, Northern Europe, West Coast of North America, East Coast as well. It's adept at getting established in disturbed sites, but it's also great at invading these grassland ecosystems. And the issue with that is that it alters soil properties as a nitrogen fixer, and it also affects nitrogen cycling, and it also sort of enhances the ability for other invasive species to join in. And so the question is not necessarily, for me, was more of an application. How can we find this? How can we identify it? And when I was looking at previous studies, most of them dealt with bloom color. And the issue with bloom color is that they've essentially already established their roots. And if they've got to here, they're going to seed. And if they seed even 1 year, the seed bank is so strong that you'll never be able to get rid of it. So the idea with this is that we could perhaps use leaf functional traits to try and identify them before they bloom and before they seed. So our question was, do differences in measured and predicted leaf nitrogen scale to the canopy level? And essentially what we did was looked at the measured and the predicted values, both of which were clearly significantly different, and then created or simulated canopy spectra using the chemical traits that we derived for all of the species, and compared with spectrum, with the rest of the site species. And you can see here this is a significant difference. And so we use the leaf level models, and these trait prediction values here are wrong, or they would not represent what's on the ground, but they do still allow you to distinguish Scotch Broom from the site. And interestingly enough, these two outliers are also nitrogen fixers. They're outliers of nitrogen fixers. So at the very least, we can show on the landscape where nitrogen fixing plants are. And so that could be very useful for land managers. This was submitted to Neobiota, but I just got an email that it's now under review when I got back from lunch. So also good. Next steps. So essentially, the idea was to use machine learning and lots of the spectral and functional trait data that we have collected to try and estimate diversity, really. And so there are a couple ways we were going to do this. The first was going to evaluate species diversity at the subplot level. We have 108 subplots and lots of vegetation data for each. And we're going to use both the spectral information and the predicted functional trait information. And then part two has to do with the stressors. And I'll get to that after I'm finished talking about part one. So here is an example essentially the spectral representation of plot 7, subplot 1 from the VisWe or HiSpecs data. And it looks great. Thank you very much, Margaret and everybody. And why is this important? Well, so you can see, even just looking at the visible region here, a noticeable change between plot 7 subplot 1 and plot 7 subplot 9. So these are less than 2 meters apart, and you can already see that in the visible region there is quite a difference. There's also one sort of around 1500 and up to 17, and I just didn't highlight it, but there's lots of variety even within a plot. So we may be able to find some metrics that could help us understand diversity a bit better. Previous presentations today have talked a lot about spectral statistics and other things that they might use. I'm looking at a couple different methods. None of this is set in stone yet, but the goal is essentially to use the data we have to try and estimate diversity. So richness is probably going to be pretty challenging. So we'll move into diversity indices. Etienne was mentioning today that Simpsons is probably a good place to start, but you know, I'll explore and see what we can find. So this is still talking about part one, and essentially what I've done here, let's talk a little bit faster, is I've taken a subplot and predicted using the PLSR models that we generated to predict a trait, and then normalized it here so you can see green is high, is pixels with high values of deep nitrogen, high values of LNAs, green over here, and high values of 8% carbon. This is just plotted in RGB. The idea being that you can quite clearly see the same or a strong distinction where there is likely fast-growing high nitrogen plants here and maybe slower growing plants of a different species or different life form, perhaps perhaps a grass or something. I don't actually know for this plot what those are, but I will soon, hopefully. And then 2, this is a functional distribution. So this is an entire flight line that was collected with the high specs. And down here is a road, up here is a road, up here is the encroaching Douglas fir. And the idea behind this is that we are going to essentially determine if there are distances from any of these stressors, or I guess if any of the traits are randomly distributed from those stressors, if they're more or less related to that. We can include some climatic or topographic variables as well, like wetness and other things such as that. And as I mentioned earlier about LNA potentially being useful for tree crown delineation, you can actually see here quite clear, and I took the slide out, but you can make out the crowns of Gary Oaks quite well from this image. That's it.